Next up in this session, we have FrederickJan van Schooten. He's from Maastricht University and heads the Department of Pharmacology and Toxicology and leads the Division Respiratory and Age-Related Health of Neutrim School of Nutrition and Translational Research in Metabolism. He's more than 35 years of senior management experiences in life sciences, research, academic education, heading departments in faculty management, public health policy. He's published over 300 papers in peer-reviewed journals, including 50 with clinical research on breath biomarker discovery and statistical handling of breath biopsy data. Over to you, and thank you.
I want to thank the organizers to invite me. It's an honor and privilege to speak here. First, I want to give a brief introduction of the work we are doing in Maastricht, followed up with introducing you a new sampling device for breath from mice. We developed this device to perform future animal studies to get more insight into the relation between breath volatiles and pathological processes. And I will end my presentation with an example of a bacterial-induced abdominal sepsis model. This is the dream team working on breath research in the Department of Pharmacology and Toxicology. As you can see, the team members have varying expertise ranging from chemistry, mass spec, data analysis, and biochemistry. We are closely working together with the clinicians from our medical center and in the scope of the present talk, the people from the gastroenterology department and surgery. The background of the researchers nicely illustrates the multidisciplinarity of our field. Here, the principal approach we use in our clinical studies. We are measuring the volatile chemicals by trapping them on desorption tubes and analysis by GC-TOF-MS. We nowadays use the receiver for collection of the breath. When all the patients are sampled and all chromatograms are obtained, the data is pre-processed, the background subtracted, peaks picked, the chromatograms aligned, and normalized to account for sample integrity. When the database is ready and linked to the clinical records, several machine learning approaches are applied to search for FOX biomarkers that can be related to disease. This can be untargeted and start from a holistic point of view, or via a targeted approach by selecting first the compounds that can be of interest from prior knowledge, and this latter is more a hypothesis-driven strategy. This slide shows the examples of our research. We are performing human studies focusing on lung, gut, and liver. To support the clinical studies, we are performing headspace analysis, headspaces from bacterial cultures or in vitro cellular cultures, mainly with the purpose to get knowledge of the origin of the compounds. From which endogenous processes are they coming from? More recently, we started this with using small animals, on which I will report later in this presentation. Breast research is a fast-growing field. A quick PubMed searching for the keywords breath and FOX shows that in the early 1990s, there were only few papers per year. When we entered the field with the first paper in 2007, the number of papers was starting to rise. And nowadays, in September 2020, already 100 papers are published. And in the end of the year, that will be something around 130. This is good to see and shows the interest of our fields. At the same time, with the fast acceleration of studies, a next level of complexity is coming up compared with the early years. Nowadays, many different clinical studies using various analytical mass spec approaches, sensing technologies, leads to a vast amount and varying results. In the early days, it was relatively easy to oversee the field, and now it is much more difficult. At present, many predictive biomarkers, disease biomarkers, drug response biomarkers, and so on are proposed. However, still it appears not easy to find the right compounds to target and base diagnostics on. An example of this difficulty is the example of colorectal cancer. In my group, we are trying to find markers in breast and feces that can detect the cancer in an early stage. Recently, the PhD students Robert and Hauran made an inventory on fox and the microbiome in relation to CRC. 71 studies could be identified when considering all matrices into account. In all matrices, different classes of fox were increased or decreased in CRC patients compared to controls. System-wide, those classes included fox related to fiber fermentation, protein fermentation, oxidative stress, products related to the TCA cycle, or products related to Warburg metabolism. For breath, 11 studies could be found. Breath appeared to be the most difficult matrix. General trends are hard to find. Studies reported on very complex branched alkanes and aldehydes, of which results are often not reproduced across groups. However, compounds that are, for instance, promising in feces, such as short-chain fatty acids, branched-chain fatty acids, and lactic acids, are also found in breath. These compounds are less complex and can be hypothesized coming from certain biochemical processes or to gut microbial changes. So this calls for systematic investigation of high potential fox from matrices besides to breath. Overall, the review shows that it is difficult to identify the crucial compounds that can be used for a diagnostic tool. And much more basic knowledge needs to be generated about the biological origins before we can enter the clinic with reliable markers. Some in-between conclusions. Volatile markers goes beyond lungs and breath. You can also target internal organs like liver and colon. Clinical implementation is difficult. Many Fox biomarkers are proposed. There are many different sampling and analytical approaches, so there is a need for standardizations. And at present, there are several initiatives in the field to tackle this. It's not very clear how specific Fox are for certain pathological processes. So there is really a need for validation studies in large independent patient groups. Elucidating the origin of Fox. This may go ahead with headspace analysis of cellular and bacterial cultures or via breath sampling of animal models. And I want to continue with that. Why animal studies? Animal studies may provide standardized disease models, for instance, transgenic or chemically induced models. Diet, housing, and other environmental influences can be controlled more easily than in human studies. The assumption is that animal studies can serve as proof of concept that there is information in breath that can indicate or predict the onset of disease. Long-term animal experiments are often limited by invasive breath collection, use of anesthetics, and terminal experiments. For instance, the anesthetic isoflurane end up in the breath and potentially influence the exhaled breath profile. Thus, our goal was to develop a non-invasive breath collection in mice using nose-only restrainers thereby omitting the need of anesthetics. We developed the device in close collaboration with Rudolf Jäger, an eminent inhalation toxicologist and founder of CH Technologies, a company dealing with inhalation toxicology and the delivery of bioaerosols, nose only to small animals, based in USA, New Jersey. Instead of delivering polluted air nose only to the mice, we reversed the system and trapped the exhaled air of mice in absorption tubes. This is a picture of the system. You see 6 glass restrainers in which the mice are placed. Here you see 3 mice with the nose in the front and the absorption tubes to collect the exhaled air. The week before starting an experiment, the mice are trained to enter and stay in the restrainers via a certain training protocol. After the training, the mice are quite at ease in the restrainers. Here are some parts of the device. The restrainers are made of glass, which is easy to clean and also known to be inert and expected to have little impact on the FOX secreted by the animals. A acrylic nose cone that is placed over the nose and connected with the sorbent tubes. And all the tubing is made of polyurethane. This picture schematically shows the sampling device. As said, the device has restrainers to keep the animals with their nose in the nose cone, where an airflow is provided along the nose of the mice. The device is equipped with a filtered air supply to provide clean air to the nose of the animals to improve signal-noise ratio. The same filter is used as in the receiver samplers that we use for human sampling, a carbon HEPA filter. There's continuous CO2 monitoring of exhaled air for indication of the rodent's well-being. The vacuum system at the end provides a negative pressure to pull the exhaled air through the sorption tubes. Flow controlling chokes determine the speed of the airflow. The most optimal flow and collection time may differ between animal species and the type of the sorption tubes that are used. This is an example of a chromatogram of a breath sample measured with DC TOF-MS. Some dominantly present compounds are indicated. The breath sample in red and the blank sample in light blue. There is much more signal in the breath chromatogram compared to the system blank. So it appears that a significant amount of FOX is added when air passes through the lungs of the mice. First, we wanted to check the system to see whether there are differences when mice are placed in different restrainer positions. We used 3 mice. The open positions are used for background measurements. Additionally, we wanted to optimize the time of sampling and the airflow. The flow is determined by the settings of the flow controller chokes. Obviously, the airflow is important in relation how fast the FOX are absorbed on the tubes. This is a general picture of all the samples together, looking at the whole FOX profile in an unsupervised manner. Blue color are TD blanks. That means sorbent tubes without any air sample in it. Trustworthy, they cluster nicely together, showing not much variation. Systems blanks are in red, exhaled air from mice are in green. You see a nice separation. Mice breath is different from the blanks, and thus something is added to the tubes and confirms that we are looking at breath. First, we looked at the influence of the different positions of the restrainers. At the left, the mice color-coded according to the different restrainer positions. You can see that it clusters with the mice, which is good. For instance, green is the same mice in different restrainer positions, and the fox profiles group together. Overall, it seems that there is not much influence of the restrainers, that the position does not matter much, is also illustrated in the right panel. When color coding according to the restrainer positions, there is no clustering. We wanted to pass a certain amount of air over the tubes, similar to the volume we sample in humans. So we had to consider the time and flow and the interaction between them. Is there an effect of combining flow and time on the total Fox profile? What we saw is that the flow has a stronger effect than the sampling time. It does not matter much between 25 or 20 minutes. There is less variation seen for the green combination, that is a flow of 239 mil per minute and a time of 25 minutes. So that will be a good setting for further experimenting. Here we looked at separate compounds that were tentatively identified in the breath of the mice. In this table, we see the different flow and time combinations and the compounds with the mean standard deviations. These standard deviations are generally pretty good. Many compounds show low variations between the mice. On the other hand, some compounds have high variations, as for instance, isoprene. That is pretty variably measured. But it is known that isoprene fluctuates a lot due to muscle activity, and probably the mice moved differently. After the initial experimenting with the device, we performed a study with an abdominal sepsis mouse model. Why this model? A fear complication in patients that undergo colorectal cancer surgery is an anastomotic leakage. In this situation, intestinal content leaks into the abdominal cavity, causing an abdominal sepsis. Obviously, this is a severe condition that requires early and adequate treatment. There is a need for markers that early indicate whether abdominal sepsis is developing. And because of its non-invasive character, the analysis of Fox in exhaled breaths would be an interesting tool. So we performed abdominal sepsis with E. coli, Enterococcus faecalis, and their antibiotic counterparts ESBL and VRE in mice. The bacteria were isolated from human feces. These bacteria were injected IP with a certain dose, and breath was collected at time points 1, 3, 6, and 12 hours after injection. Also, blood was collected at 6 hours and 12 hours. After peritoneal injection, all animals developed an abdominal sepsis, starting with local inflammation that later evolves in a more generalized sepsis. In the plots, we see IL-6 levels that were increased and shows that inflammation was induced. When observing the animals, we could see that sepsis was occurring based on symptoms. And it is still work in progress to quantitate the sepsis by calculating a so-called Murine sepsis score. Are we looking at breath? looking at all samples in an unsupervised manner. In this plot, samples for mice are in green and blank samples are in red. And we see that breath clearly differs. So yes, we are looking at breath, and there is something added to the exhaled breath from the mice compared with the inhaled air. Is there a difference in breath for mice treated with the respective bacteria? In this plot, the E. coli treated mice in red, its antibiotic counterpart, ESBL, in light blue, E. faecalis in green, and its antibiotic-resistant subtype in purple. We see separations between the groups. The Fox profiles for mice injected with E. coli differs from the E. faecalis. Which compounds are the drivers between the separations have still to be elucidated. This is work in progress. The time trajectory for the mice injected with E. coli, as an example. In dark blue, the samples obtained at T0 and T after 1 hour. After 6 hours, you can see the Fox profile moving to another place in the plot. After 9 hours, to another place. And after 12 hours, again to another place. Therefore, it seems that the volatiles are changing accordingly to the developing sepsis. Also here, we have to figure out which compounds are behind this trajectory and link them to the accelerating sepsis, which is work in progress. Now I come to my summary. Sampling of mice. We have developed a nose-only approach. 5 mice can be sampled simultaneously. We use filtered air to reduce background compounds. Various compounds were detected in the breath of mice. The sampling device was successfully applied to study abdominal sepsis. We see changes in the Fox profile in the breath, and there is a distinction between E. coli and Enterococcus faecalis. The next steps we have to do is to identify the crucial compounds and try to explain biologically. Now I have come to the end of my presentation, and I'm happy to take questions.
Thank you so much for the wonderful talk. Really appreciate it.
Okay.
I totally agree that having good preclinical models, in vitro and animal models is really important for the field.
Yes.
So few questions just to kick us off. So the first one in was, do the mice experience any stress in the restrainers, and could that affect the VOC profile?
Yeah, that's a really good question, because stress can affect the metabolism of the animals, of course. And we trained them prior to the experiments. So we started with that 1 week before the experiment and trained them for the days, and starting with putting the glass restrainer in the cage so they can sniff to that and become acquainted with it. And then we put them in the restrainers for a few minutes, let them out, and then do that several times a day. And then over the days, increasing the time in the restrainers to 25 minutes. And actually, they are quite comfortable at a certain moment. Sometimes they even go, let's say, voluntarily in the restrainers. And then in the end, you do the whole system by to mimic the experiment without measuring anything. And we have the idea when the mice are back and so on, they are behaving quite normal, and that the stress level is not that high. Mm. So it's a good question. Yeah, so we train with a certain protocol.
That's great, thank you. And I suppose that the great thing about the setup as well, it makes it much more easier to do longitudinal measurements compared to
bases. So they, of course, they keep alive, and you can just measure all kinds of time points and see whether certain disease is progressing. Yeah.
Fantastic. I had a quick question related to the setup in the background. So having the glass materials, you showed that you had low VOC background, the system blanks. Looked like there was good signal originating from the mouse when you looked at the raw chromatograms, and you showed a few of the example compounds and the variability of those compounds. I was wondering if you were able to get a bit of a sense of how many on-breath VOCs there were when you compared the mouse breath to the system blanks. I know there's the table with a few compounds in there, but do you have a sense of the total number you were seeing on breath?
Yeah, typically hundreds of compounds are measured, and 200, 300 compounds. We have not identified, of course, these compounds. So as I showed in the presentation, we identified 27 compounds, which were quite abundant and not present or very low present in the system blanks. And yeah, these compounds are just coming from endogenous metabolism because these were untreated mice. But yeah, these were this oxidative stress like pentane, hexane, or aldehydes. You could hypothesize that. Or we also saw xylene, or let's say toluene, which then is known as air pollutant. So probably they have picked it up earlier and stayed in the system, and they exhaled it again. And ethanol was detected, for instance, which could be related to gut microbial changes. And yeah, the challenge is, of course, to see whether these compounds are changing during a disease development. And we're still in the progress to do with the abdominal sepsis to identify the compounds and the changes we see. And then if we have nice results there to translate it to patients, we are collecting patient materials from patients that underwent colorectal cancer surgery. And some of them do develop sepsis. And then, yeah, the big challenge is, can we translate the mouse model then also to the human situation? Yeah.
Yeah, that makes sense. So you'll confirm the idea in the animal model and then run the targeted methods for those human subjects?
Yeah. That's the plan, yeah.
That's very nice. That's very nice.
Yeah.
So of the table, they were all confirmed IDs. And then what was interesting was a few were jumping out that we know are present on human breath as well. So it would be really interesting to see a translation question.
Yeah.
Great. Let's have a look at some of the other questions. Was the breath air provided from an ambient room or specialized clean gas?
No, it's from the ambient room. Yeah, so it's not prefabricated air. So it's from, and then it goes to, it's actually the Casper system for what we use in the receiver device. That was the question. Yeah.
So is the air scrubbed that's going to the animals?
Yes, that goes to the animals, then to the nose, and we keep the pressure, and that there is a normal pressure, so we control that. Yeah, so then.
Great. Okay. Are you, this is an interesting question. I think the nose cone may help here. Are you able to check that you truly measure breath and not VOCs from skin, hair, etc.?
That's a good question. In the past, but it was years ago, we tried to measure breath from mice just in cages, and then let's say take the headspace of the mice, so they were then of course exhaling, and then you see that, but then of course then it's contaminated with fur or with feces or whatever. So then we thought, well, we need a nose only. So I think I'm pretty sure that's coming from the breath. Yeah. So there's no contact the air with the with the fur or from with the animal. So I'm pretty sure it's coming from the breath.
Is that because the animal is pushed up into the nose cone?
Yeah, to the nose, and then it passes through the nose, and then it's immediately going onto the tubes.
Yeah.
So yeah, I think it's pretty sure it's coming from the breath.
Got it. So you mentioned that you're identifying the VOCs in the sepsis model. Is there plans to do any other animal models?
Yeah.
Some of the other, the hepatology research you do, for instance?
That's a really good question. I really would like to apply to colon cancer models of mice, and then, for instance, use mice that are knock-in or knock-out mice, for instance, the APC min mice that develop spontaneously polyps and tumors. That would be really nice. Or chemically induced models in which then you can also mimic colitis-induced colon cancer, and then following it up and see whether you can get early warning signals, and that will be really something if you can show that in animal models. And then the question, of course, is if there are compounds that you can use as warning signals, whether then it's applicable to the human situation, that's of course the question. But just to show in an animal if there are tumors, are they coming up in breath? That would be a very good proof of concept.
Brilliant, thank you. Next question is, isn't 3.6 liters saturating 1 TD tube? And also, what QC are you using?
The QCs in the measurements, and what we are doing is using deuterated compounds. We adding them to the tubes and then doing the whole measuring schemes, putting in cast mixers in between. So that's what we're doing is for the analytical side.
And the other question related to the breakthrough volume, so the 3.6 liters.
Yeah, that's we choose the flow rate because it's more or less similar as we use in the human situation, 200 mil per minute. That's the TNX tubes, more or less what you use. So we try to use that, and then 20 minutes. That was also an educated guess. Probably we can do less, but we did not experience that the tubes were overloaded.
Brilliant, thanks. Time for one last one. So you talked about some other potential models, colorectal model. Is there been any thoughts or plans about where you could administer a substrate to the animals?
Yeah, that's a good one, because I saw the previous talk, of course, from Alexandra. It would be really nice to apply EFOX, similar to what Alexandra is doing, and see whether, yeah, certain diseases are then metabolizing or changing this EFOX, and then study the kinetics of the exhalation in the breath. That would be really a nice approach to apply. Yeah.
Brilliant, excellent. Well, thank you so much again for the time today and the talk. Really appreciate it. Thank you.
Yeah, it was a pleasure, and a really nice conference. Congratulations with that.
Thank you. Glad you're enjoying it. Thanks again.
Yeah. Okay, bye-bye.
Bye.